Alright, last video for chapter 2, so part 3 of 3. So this last section focuses on organic compounds. So organic compounds are compounds that use carbon as its base, and then we add to it to make different molecules. So we have 4 different categories of our carbon compounds. We have carbohydrates, lipids, and proteins. All 3 of those are considered macronutrients, so things we need a lot of during the day. That's why we eat so much. And then the last section are your nucleic acids, which are your DNA, RNA, and ATP, and a few other nucleotides. So for our organic molecules to work, we have our carbon atom base, which has 4 valence electrons. So in that last outer shell of that atom for carbon, it only has 4 electrons. It likes to have all 8, just like other atoms. So we have 4 empty spots to create a bond. So this is how carbon makes bonds. So carbon does like to bind with itself a lot. So here we have this carbon chain here. This is a simplified version of what's going on. It's kind of one of those things where you understand that each of these little peaks is where there is a carbon atom, and that you also just automatically, you just have to know this, understand that a hydrogen is attached to it. So same thing, this little random zigzaggy line, each little point of the little zigzag, that is where there's a carbon molecule, ending with that last carbon there, and that one would be a methyl group, really. And it has 3 carbons to make carbon atom happy. So it can make chains, it can make different branches, it can make a ring and just bind with itself. These 2 dashed lines mean that it is a carbon double bonded with itself. So here this carbon still has its 4 molecules, 4 bonds, so with hydrogen, another carbon, and then double bond it with another carbon to make 4. So when carbon makes bonds, it creates covalent bonds, so it's going to share the electrons, and it typically shares electrons with hydrogen, oxygen, nitrogen, and sulfur. So these are common atoms that you'll see in many organic molecules. So for these carbon backbones, they can carry functional groups that are going to determine what kind of molecule it's going to be. So here we have different functional groups. So up top we have this hydroxyl group, so an oxygen and a hydrogen that's going to attach to that final carbon of a chain or other structure. And this is what creates our sugars and alcohol. So that little group on there is going to be the functional group that creates the attributes of that molecule being either a sugar or alcohol. Methyl groups, so that little long chain I showed earlier, this is the last end of that long chain. You'll find that on different fats and oils, as well as amino acids. Carboxyl group, so it's a carbon double bonded to 1 oxygen, and then an oxygen and a hydrogen on the other side. You'll find that a lot in your amino acids, some sugars, as well as in proteins. You'll see a carboxyl group. Amino group, nitrogen, 2 hydrogens, so an amino acid, a bunch of amino acids, you put them together, that forms a protein. And the last group is a phosphate group. So we're going to see that one a lot in different structures. Our ATP molecule for energy storage. So we have our phosphorus here in the center, 4 oxygens around it, and then those hydrogens off the side of that. So making up our nucleic acids, and one of our energy molecule, ATP, actually has 3 of those types of phosphate groups on it. Okay, so putting together our different carbon molecules, you actually put a bunch of them together. So we have like a monomer, which is our 1 unit, mono for 1. Put 2 together, there's a di equals 2. Oligos, a few more. Polymers, even more. Macromolecule will be something like on the size of DNA. So when you keep adding those building blocks to each other, it creates what's called polymerization. That's the process of joining them together. So in order to do this, the process is called dehydration synthesis. So we're going to dehydrate something. We're going to remove water off these two monomers, these two building blocks. So when that water is removed, we've created a oxygen molecule. This leftover oxygen is going to be the, let's say bond, but the connector between our two monomer building blocks, which is now a dimer. Hydrolysis is the opposite effect. So instead of removing water, we're going to add water to break apart this larger molecule. So this is how we use digestion, breaking apart our food, so we can actually extract out the energy and get it to a form where we're able to absorb it into our body. So our water ionizes into a hydroxide ion and a hydrogen ion, and that helps to break that bond between our dimer. Next. Okay, let's focus on our organic compounds, carbs, lipids, proteins, and nucleic acids, which will end off our chapter. Yay! Okay. So carbs are hydrophilic. They like water. So examples of that be sugars and starches. So of course you know that from just nutrition and stuff. So your generic formula for a carbohydrate, no matter how many carbons it's going to have, for every one carbon, it's going to have 2 hydrogens and 1 oxygen. So with our carbohydrates, we start off with our monomer building block. So a mono meaning 1, and a saccharide, so sac really means sweet. So our 3 really important ones are glucose, which is blood sugar. Sorry, can't see that. Glucose, which is blood sugar, galactose, and fructose, which is the sugar found in fruit. So with glucose, you actually can take that as not the slide for it. We'll talk about that later. Okay, so we take 2 of those monomers, we put them together, we create a di meaning 2, a disaccharide, put them together, and we create sucrose, which is table sugar, lactose, which is the sugar that's found in milk. So people who are lactose intolerant, they've lost the ability to break down lactose. So we're able to do that as a child, as a baby, when we're still nursing. And then over time, we stop nursing, so there's no point of us still creating that lactase enzyme, and so we no longer are able to break down that lactose milk sugar. Some people, they maintain the ability and creation of that enzyme, and so they can have their dairy products, and they're fine. Okay, last one is maltose, which is found in grain product. So you keep adding on additional monomer carbohydrate building blocks, and you can form an oligosaccharide or polysaccharide. So these are actually more important, so we're going to focus on those. So we have glycogen, which is a bunch of glucose stuck to itself, and that's the storage molecule for glucose is glycogen. So different cells will just have their own storage for energy, particularly our muscles, which always need energy, our brain, which is just stingy. Our brain only uses glucose as a energy molecule or carbon molecule to gain energy from, and so it will have its own sugar storage. So it will make you eat if your blood sugar is low. That's why you get all cranky. The uterus, as well as the vagina, also stores glycogen, and that's because the bacteria there will break down this glycogen and create an acidic environment that way because it is open to the outside environment. That acidic environment will help to kill off any kind of foreign invaders, kill off weak sperm, and that way just adds a protection layer for those two. Another polysaccharide is starch. It's energy storage for plants, so the glycogen version, but for plants. And we're actually able to eat and break that down and use that as an energy source. The last one is cellulose. Cellulose is another carbohydrate storage molecule in plants. So it's more structural. We don't have the ability to break it down. We can make it to smaller bits of cellulose, and that creates our dietary fiber, also called roughage for some people. They have, grandma probably used to call it roughage. And that just helps to keep our bowel movements and our digestive system keep going and flowing versus you end up with stoppage because you don't have enough fiber, and then you end up with constipation. On the other hand, you actually can have too much fiber, and that can cause some problems too. So most likely, unless you're on a vegetarian or vegan diet, you're most likely going to be on the need more fiber than the need to cut back on the fiber group. Okay, so here's our glycogen finally. Sorry, should have put that earlier. So each of these is a glucose molecule. We dimerize or polymerize a bunch of glucose molecules, stick them together in this branch structure called glycogen, so we could store glucose into the form of glycogen. I'm going to add that to that slide. Okay, so what's the point of carbohydrates? Why do we need them? Energy. That is our main source of using energy in our body. And then we can also break some of that down and form different structures. So we have our glycolipids, which is part carbohydrate, part lipid. Glycoproteins, part carbohydrate, part protein. So both of these are on our cell membrane. We'll talk about that in chapter 3, and also helps our body recognize a cell that belongs to the body versus a cell that does not. So that's actually, we get into that really deep on chapter 21, but that's AP2 material. Proteoglycans, which is mostly protein and then a little bit of carbohydrates, and that's also another structural situation in our body. Okay, so this is going through all those different carbon organic molecules, those carbohydrates that we just mentioned. Okay, moving to lipids. Lipids are hydrophobic. They don't like water. Lipids do have more calories per gram than carbohydrates, which means that they are a more efficient molecule for energy storage, which is why we store more fat than we do anything else. So there's 5 different types in humans. We have our fatty acids, our triglycerides, phospholipid, ecinosoids, and our steroids. So our fatty acids, big long chain of lipid. Some of it we do have to obtain from food. Some of it, our liver, which does way too much, can actually create stuff. So create some of the ones that we don't need to take in through our diet. So we take 3 fatty acids, we join them to a glycerol molecule, and we create a triglyceride. So tri for 3, as in that's how many fatty acids are linked to that glycerol. So this is what we use to store in our fat cells in order to store that excess energy. So if when you, in between meals, when you are unintentionally fasting at night because you're asleep, our body can, once we get to a certain level with blood sugar, start breaking down the fats. So again, primary function is for energy storage. So that's just putting those 3 together to show the difference between them. There you go. Keep going. Okay. Another type of lipid is a phospholipid. So phospholipids, very similar to a triglyceride. So we still have 2 fatty acid chains. Here's our glycerol. But instead of a 3rd fatty acid chain, on the top we have a phosphate group. So this phosphate group is hydrophilic. So this part is what's called our hydrophilic head. Here is our hydrophobic tail. And this is actually what makes up our cell membrane. So we'll talk about that structure more in our cell biology chapter, which is chapter 3. So we have all these hydrophobic areas. They're going to point toward more aqueous or water environments of the inside and the outside of the cell. While these hydrophobic regions, they'll stick together and will form that barrier. So hydrophobic head region, and then this is the hydrophobic tail region. They will actually point towards each other. Okay, I think this is last section on lipids. So we have a cholesterol, so the stuff you get from eggs or other animal meat products. That is considered the parent steroid, where we can make other steroids. So, for example, your estrogen, your testosterone, those different types of hormones, bile acids help to break down your fats. All of those are made from cholesterol. Most of our cholesterol can be made by the liver, so we don't need to eat that much. Depending on how much comes from your diet, the rest your liver can make up, and your liver will just adjust. So, let's say you had a Saturday morning, you had French toast, you had scrambled eggs, had some bacon. That's a lot of cholesterol all at one time. So your liver's not going to make as much cholesterol for a moment. Okay. Proteins. Keep going. Don't ask why this is animated. Oops. Okay. Protein. So a polymer, so multiple building blocks put together of a monomer. So our monomer for a protein is an amino acid. So there's different portions of an amino acid that all amino acids have. You have an amino group, that's where it gets its name, a carboxyl group, And then the radical or R group, which is what determines the type of amino acid it is. So we have 20 different types of amino acids, and all these different types of building blocks are what's going to be used to make a protein. And the type of amino acid, again, is based off that R group. So we have some proteins that have hydrophobic regions. We have some proteins that will have portions that are hydrophilic. So that's actually going to help with its function. How is it inserted into a cell membrane? How is it going to let stuff in and out of a cell? So a lot of times it's actually related to its function. Okay, so this example is a different amino acid. So all this being the same, that pink being the R group that determines what it is. So in order to join these amino groups together, we do so with a peptide bond. So a peptide bond is made by dehydration synthesis, and that's what's going to join our amino acids to each other. So we'll actually go through that process during the cell biology chapter. So we put two amino acids together, di for 2, tri for 3, oligo a little bit more, poly a bunch, and then once you get to a chain of at least 50 amino acids, then it's fully called a protein. So as you're breaking down protein in your diet, it's actually going to go back up this way. So it's not like, okay, let's start stripping off amino groups. You actually break it apart into smaller bits, get down to dipeptides, and then you finally can break it apart into amino groups. Who knew? Okay, so that's just showing you how to make a peptide bond. So with proteins, they start off as just a chain of amino acids. Try to make this a little bit better. And you then fold that protein up into its conformation. So its conformation is the unique three-dimensional shape of a protein. So it's the shape it needs to in order to have its function. So necessary for muscle contraction, enzyme being in the correct shape to do its job. So we'll talk about that in chapter 3. Channels inside of a cell so you can allow stuff in and out the cell. So you can have a conformational change where, let's say, if my hands were a enzyme and it can change shape to grab onto something. I'm not going to break this, but also change shape in order to, let's say, if I was going to break down this paintbrush. So my enzyme protein would change shape in order to break that. So you can have some kind of conformational change and cause where the protein won't lose its shape. Now, if it's an extreme one called a denaturation, that's where it's going to destroy the function of that protein. So this is caused by either extreme heat or pH. So this is why you have to maintain your body's correct body temperature as well as your body's pH, because it can affect your proteins. They don't work, your body doesn't work. So for example, when you cook an egg, you add heat, you get wonderful scrambled eggs. So we're taking all of these proteins here, we're actually linking them together, and that's what creates that, even if you're doing egg whites, creating that linkage and totally changing the shape of that protein. So with our proteins, we have different structures. So we start off with our string of amino acids. That's the primary structure, either folding it up into a coil or a sheet. That's the secondary structure. Tertiary, so it's third level. It's some straights, some curly, some beta sheets. So just again, depends on the function of the protein. So this is the last final level of protein structure of a individual protein. Now you can have the last section, which is quaternary structure, which is where you have multiple proteins getting together for some kind of function. So for example, hemoglobin, which is found in your red blood cells in order to store, be able to bond to oxygen. You also can have carbon dioxide binding to the protein portion of hemoglobin, while oxygen binds to the heme portion. So we have 4 different proteins that create this quaternary structure. So tertiary is the last level for individual protein, quaternary for a multiple proteins working together. Okay, so there's different functions for protein. Let's skip all that. Okay, enzymes. So enzymes are usually going to have some kind of job. So their naming of it is ACE at the end of whatever they're breaking apart. So for example, amylase, ACE for enzyme, breaking down amylose or starch. For example, lipase, ACE for enzyme, and the lip part of lipase for the lipids that it's breaking down. So the job of enzymes is to lower the activation energy. Basically, it's going to make your reaction a whole lot easier and faster. So here we have two different reactions looking at the energy needed for them. So the reaction on the left is one that does not have a enzyme. So here we have our starting point for our level of energy. We have to put a bunch of energy in. So for example, move the molecules around so they just happen to bump into the right binding so that they can have their reaction. So you got to put a bunch of energy in and then the reaction will go. Well, if we have a catalyst or a enzyme in order to speed up our reaction, it lowers the activation energy. So you just need a little bit just to get everybody in their spot, in the correct places, so that the enzyme can do its job. So same net energy released for both. For the reaction, it's just a whole lot faster because you lower the activation energy that is needed for that reaction. So with our enzymes, I said move. They can actually be reused. So here we have an enzyme. It's going to work on something, a substrate. That substrate and the enzyme fit in a lock and key type of situation. So, for example, our amylase isn't going to break down lipids. It's just wrong shape for it. It's shaped for it, so it's not going to do the job. So again, why we need to have our proteins being in the correct conformation or correct shape that it needs in order to do it. So here we have our enzyme. We go through a conformational change to hold our substrate into its position. The enzyme does its job, and then it has another conformational change to release it. Now this enzyme is able to do it again, so it actually helps even more speed up the reaction, and you're able to go over and over again and use it a lot in order to finish off your reaction. Okay, so reusable, lots of speeds. Okay, last carbon molecule is our ATP and different nucleotides. I want to quickly do this one just because we cover this in chapter 3 as well. So a nucleotide has a nitrogenous base, a sugar, and at least one phosphate group. So a very important and well-known nucleotide is adenosine triphosphate, or just ATP for short. So it has a adenine for its nitrogenous base, a ribose for its sugar, and 3 phosphate groups. So there's the triphosphate portion. And that is for its energy storage function that it has. So we can break off bonds with these phosphate groups, the first and second one actually, and be able to release it for energy. If you do both of those, this little phosphate group is going to wrap around, connect back with the ribose sugar, and create what's called cyclic adenosine monophosphate, so mono for one. And that can actually be used as a signaling molecule in the body. So ATP's job is for energy transfer. So you store energy into the ATP molecule, and then wherever you need energy, you break that bond, you release the energy. Okay, so in order to do this, you have an enzyme called ATPases that's going to break down, so ACE to work on the ATP molecule. So go ATP, so 3 phosphate groups on there, break off one, you go to ADP, touchscreen. Oh no. Okay, so go to ADP, so 2 phosphate groups, and then you have a phosphate just hanging around in the cell. It'll get reattached to an ADP or whatever the cell might need it. But that breaking of the bond releases the energy. Opposite situation is called phosphorylation. So the enzyme that does that, ACE for enzyme, are kinases. Those kinases will add phosphates to ADP to go back to ATP. You also can do it to turn on and off a protein. So that's going to change its shape a little bit and determine determine whether or not that protein's going to function or not. So sometimes you don't need a protein to function at that moment, and so you can turn it off and on. So with our ATP, we'll go into more detail for this for AP2, but you have your specialized organelle or little factory in your cell called the mitochondria. Its job is going to make ATP. Your body can make without you having the mitochondria, and if it does so, it's called anaerobic fermentation, but it only makes 2 ATP molecules per every glucose. But you get your mitochondria, your powerhouse of the cell. Your production method is aerobic respiration, so you need oxygen. There's your aerobic kind of huff and puff. Need the oxygen, and you actually can make 36 ATPs in total through that process of breaking down that one glucose molecule. So a whole lot more efficient at creating ATP. So there's other nucleotides, I'll skip that. So we take our different nucleic acids. We'll talk about this more in chapter 3, but you can do you can have DNA, which is a lot larger, RNA, which is so DNA has our instructions for making proteins. RNA, we can take a copy of that individual instruction to make the protein, as well as the other types of RNA are used in order to create the structure in order to make our proteins. And that is the end of chapter 2. So remember, ask questions if you need it. Finish up that study guide. Everything you need for the study guide is in your PowerPoint. See you next time.